Hello everyone, my name is Enio Gjerga and today I will be describing Linda as a method which is used for characterizing alternative splicing effects on protein interaction networks. So, alternative splicing is a fundamental process occurring during gene expression, and it involves the merging of different regions of precursor RNA molecules, thus resulting in the production of multiple mature RNA transcripts from a single gene. Alternative splicing effects can, for example, result in the inclusion or exclusion of specific exons which encode for protein domains. This process can then generate protein variants with different combinations of domains which can alter the overall architecture of proteins and thus affecting not only their functions but also their interaction properties. As such, alternative splicing appears to be an important modulator of signaling pathways in cells. Nevertheless, despite their relevance, alternative splicing effects are typically neglected when it comes to the functional and especially mechanistic analysis of signaling pathways. To address this gap, we have developed Linda as a method which integrates information from prior biological knowledge on protein interaction and transcription factor activities with RNA-seq data in order to infer contextualized cellular networks. So as we can see, Linda requires the input from various methods and resources and about which we can discuss. So first of all, we have to keep in mind that Linda uses causal reasoning principles to infer regulatory networks upstream of significantly regulated transcription factors. In other terms, significantly regulated transcription factors will serve as the bottom layer of signaling, and Linda will aim to infer upstream protein interaction mechanisms leading to the regulation of these downstream transcription factors. As such, we need to first identify potential transcription factor candidates which appear to be regulated in a certain context. For this, we will rely on Dorothea, which is basically a resource of transcription factors to gene relations, and then we can use statistical testings to identify significantly regulated transcription factors based on evidence from, let's say, differential gene expression data. Then these significantly regulated transcription factors can be used as points from where upstream protein regulatory processes can be inferred. Another resource that Linda relies on is Digger, and which represents a large resource that integrates protein-protein and domain-domain interactions into a joint graph for humans. The interactions present in Digger resource will serve basically as a backbone or parallel-knowledge network from where Linda will identify a subset of interactions which appear to be functional in a specific context. Digger additionally maps interacting residues to exons and transcripts, thus allowing to better assess the functional consequences of alternative splicing. Additionally, Linda can integrate results from various methods which are used to identify differential splicing events, such as RMATS, MAGIC, or Baltica. And such methods apply different strategies to identify differential splicing. However, what most of them seem to have in common is that for each splicing event, they provide metrics such as inclusion level difference and the p-value. With inclusion level difference is typically referred to the change in usage or inclusion of a specific exon, while the p-value represents a significant score associated to such an event. Since Digger additionally provides mapping of transcripts to domains, besides differential skipping events, we might as well use information from differential transcript analysis, and in this case, instead of using inclusion difference values, we can use estimated log-fold change values for each transcript. Another thing that we need to keep in mind here is that as multiple exons or transcript IDs can map to a single domain, the size effect score of a domain are then simply calculated as the average among the size effect scores of their matching exons and transcripts. On the other hand, the significance p-values to the domain size effect score is determined by performing a Fisher aggregation procedure over all the p-values of the corresponding exons and transcripts which map to this domain. Finally, Linda will then make use of all these three available resources in order to mechanistically characterize splicing effects in protein interaction networks. For that, Linda will rely on an integer linear optimization problem, or as ILP that I will refer from now on. And this ILP formulation optimization problem is tailored to reconstruct networks from Digger that represent regulatory mechanisms leading to the regulation of differentially expressed transcription factors, while also, of course, taking into account the effects of domain skipping from alternative splicing. In order to give a better insights into the inner workings of Linda, we can refer to this network here depicted on the left. And here we can notice a few elements on this network. So first we can observe a set of upstream regulators, I, as depicted in grid node, and which represent the protein from where signaling propagates downstream. In this case, when the source of signaling such a receptor is known, then the users can define the input set by giving the name of the proteins. Otherwise, when the source of signaling is not really clear, then Linda adds to Digger an auxiliary input or perturbation node or protein which is connected with those proteins in the resource that do not have any upstream regulator. Additionally, we define with T the set of regulated transcription factors, and these will be at the bottom layer of the signaling network which Linda will infer. And with P are the set of intermediate nodes or proteins between the I set and the T set here, and these are all proteins which will be inferred by Linda. As we can see, some of the nodes here which correspond to proteins are made of several parts here, structural units, which basically correspond to domains. Besides nodes, we can also identify the interaction sets. So with E, we define the set of protein-protein interactions present in the prior knowledge, as depicted with solid lines. With E prime, we define the set of domain-domain interactions. And with Phi, we define the set of domain-domain interactions affected by domain skipping. So if this domain here is skipped, then also the interactions involving this domain appear to be affected by splicing. Again, we can identify a domain as skipped when its size effect has been estimated to be negative and its associated significance p-values score falls below a certain threshold defined by the users. By default, this p-value threshold is set to 0.05. Finally, we also define the set of variables in which the ILP optimization problem of Linda tries to infer. So we define with X the variables indicating the presence of a node in the solution. A node can either represent a protein or a domain. With Z, we define the variables indicating the presence of a domain-domain interaction in the solution. And with Y, we define the set of variables indicating the presence of a protein-protein interaction in the solution. So if there is at least one domain-domain interaction present, then there will be also a protein-protein interaction involving the two sets of proteins. Now, as we can also notice here is that all the variables which we try to infer take discrete values either 0 or 1, which make this optimization problem fall into the special class of Boolean or logic modeling. So in the end, the ILP formulation from Linda will infer signaling regulatory mechanisms which lead to the regulation of downstream transcription factors while also accounting for the structural architecture of the proteins mediating the signaling. So there are two modes in which we can basically use Linda, two main modes. So we have the splice-aware mode, which basically takes into account the information from splicing effects. But what I always suggest to users is to also run splice-unaware mode of Linda, which omits information from splicing effects. And I recommend doing it this way because then we can integrate in one network both the splice-aware and splice-unaware solutions, and then better observe how alternative splicing appears to be affecting, modulating, or rerouting signaling mechanisms within cells. Then for splice-aware running modes, there are also two modes in which the users can run Linda in the splice-aware analysis. So we have the hard constraint and the soft constraint modes. and the differences between the two will be made clear in how we define the objective function of the IOP problem. So this is in regards to the objective function, this is the main part of the IOP formulation, and this is the equation representing the objective function for the hard constraint mode, and the defined goal here is the identification of subset of functional molecular interaction which best reflects the evidence from differential transcription factor activities and the observed differential splicing events. As we can notice, the objective function here consists of two parts. So the first term, it says that the network solution must include as many of the top regulated transcription factors while penalizing the inclusion of the transcription factors which do not appear to be regulated. So if we provide to Linda a list of, let's say, 300 transcription factors with their enrichment scores, and we want to consider as significantly regulated the top 20 of them, then Linda will assign a score to each of these transcription factor of -1 if the transcription factor belongs to the top regulated set, and 1 otherwise. In this case, since this is a minimization problem, the network solution which Linda will aim to provide will tend to include as many of such regulated transcription factors while penalizing the inclusion of unregulated transcription factors. Then we also have a second term here for the hard constraint objective function, and this represents the size penalty term, which basically makes sure for our method to give us the network model with the best quality, and with not only with the best quality or with as many regulated transcription factors in it, but also with a minimal number of components or number of domain-domain interactions. And for the hard constraint approach, this lambda2 value remains fixed and positive as defined by the users for all the domain-domain interactions. And regarding the soft constraint approach, so the objective function here is more or less similar. However, this second term of the soft constraint approach differs a bit, and lambda2 here now represents a continuous, not fixed value, whose score depends on the estimated splicing effects. So the inclusion level values and their associated significance p-values for each domain in the prior knowledge network. So this lambda2 value will be defined by this minus log p-values. So if a domain has a negative inclusion level score, then we will apply this penalty term. So the smaller the p-value that it corresponds, then the higher the penalty. So the more evidence we have that a domain is skipped, then the higher will be its penalty term here, and the less likely is for the ILP to include this domain in the solution. And as we can see here, we have no brute hard set p-value thresholds on what to consider as a skip domain. Now, for a comprehensive evaluation, we have applied the hard and soft constraint Linda to a panel of 54 short hairpin RNA or shRNA depletion experiments in hepatocellular as well as chronic myeloid lymphoma cells from the INCORE initiative. And so we used this application to this large resource, data resource, in order to perform experimental validation of splicing effects predicted by Linda, as well as to perform computational benchmarking and to compare Linda to other comparable methods which use causal reasoning strategies. So for the experimental validation parts, we have chosen to apply Linda in two modes of analysis, splice-aware and splice-unaware, by focusing on the age and RNPK side knockdown on the hepatocellular carcinoma cell lines. And 100 diverse solutions were identified on the splice-aware and splice-unaware networks. And these solutions were then combined in order to generate a network containing 116 domain-domain interactions among the 57 unique proteins. From these 81 domains of the 116 domain-domain interactions, 27 were identified as skipped after splice-aware analysis. So basically, here is how a typical combined splice-aware and splice-unaware network solution looks like. So here, with the more yellow the interaction, the more likely is that a network has been appeared on the splicing solution, and the more purple, then the most likely an interaction appears on the splicing solution. We also have labels for the nodes, so we have with the green the upstream node, with purple the inferred protein, with red the regulated transcription factors, and also here with the blue we have the interacting domains. And then if a domain has brackets, black brackets, then this domain is considered to be a splice. So also the interaction types can either be normal or spliced, as depicted here by the dashed line. Here we took a subset from this large network, and we have noticed an interesting difference in our comparative analysis of the splice-aware and splice-unaware networks for the AGNR-PK perturbation case. And what has been noticed here was that the interactions of the MAPK3 downstream to transcription factors AGNF4 and SREBF2 appear to be disrupted when accounting for the splicing effects, as we can notice here. And the main cause of such disruption was because of the helix loop DNA binding domain of SREBF2 and the hormone receptor domain of AGNF4A, while the domain of MAPK3 appears to not be affected by splicing, so these interactions are disrupted by these two domains. So then, in order to validate these predictions, we developed AGNR-PK from HEP2 employing small interfering transfection on cytoplasmic and nucleic compartments. While depletion of AGNR-PK had no effect on the MAPK3, we observed an isoform change for SREBF2. Furthermore, the main isoform of AGNF4 is reduced on upon AGNR-PK depletion. And since AGNF4 is mainly localized in the nucleic compartment, we went to perform a co-immunoprecipitation to see the effects of the MAPK3 to AGNF4 interaction on such a compartment. And here we indeed observed that we have the interaction between MAPK3 and AGNF4 was found to be reduced upon the AGNR-PK depletion. And this observation was in line with the predictions from our network Linda models. So as conclusions, Linda was described as a method that refers regulated protein interactions based on evidence from gene expression and alternative splicing. And we performed also computational benchmarking, which are not shown here but that are in the paper, as well as validated experimentally some of the predictions made by Linda. Linda, we need to keep in mind, implements three types of solvers: Cplex, CoinCBC, and LPsolve. However, we mainly maintain it for Cplex. And it has also been implemented as an R package and is freely available on GitHub together with examples and Anchor applications. Finally, I would like to acknowledge Dr. Isabel Norman de Vries for the experimental validations and Professor Christoph Dieterich for supervising the project, as well as our funding resources. Thank you very much.